Hello students, in this lecture video I'll be discussing about the structure of contractile proteins. So let's get started. We know by now that the thin filament is made up of actin and the thick filament is made up of myosin. So let us first discuss the structures in detail. This diagram, A part is showing the actin filament or the thin filament. It is composed of three components. One is this globular or filamentous actin. Then this rope-like actin is referred to as the tropomyosin. And this one, on the site of tropomyosin, there are certain molecules called as troponin. Now these troponin basically mask the active binding site which is present on the F actin, so that myosin generally doesn't bind to it. When muscle contraction will happen, then there will be a change in the environment and this troponin will be relieved and the active sites on actin will be revealed so that the myosin can bind. Next is this thick filament, which is also called as meromyosins. They will polymerize to form the myosin polymer. It consists of one head and a tail. Head contains actin binding sites as well as ATP binding sites. So this forms actually the heavy meromyosin. This tail consists of light meromyosin, and in between the cross arm, or you can say the linkage, is called as the cross arm. Now let us go through the mechanism of muscle contraction. The mechanism is explained by sliding filament theory, which suggests that the thin filaments, they slide over the thick filaments. Now how this process starts? Basically, it is initiated by a signal which is sent to the central nervous system through motor neuron. Motor neurons are in connection with the muscles, and that junction is called as the neuromuscular junction or motor end plate. Now, any neural signal that is reaching this junction will release neurotransmitter that will generate action potential. Now, this spreads through the muscle fiber and it causes the release of calcium into the sarcoplasm. Because of this calcium, this troponin, which was masking, remember, the site on actin filament, this will relieve this troponin, it will bind. So now the masking of the active site is now finished, means the sites are revealing here. So actin will now bind to myosin. Energy of ATP will be utilized by ATP hydrolysis. Now let us see here how it is happening. This is actin filament. Here ATP hydrolysis will happen, and after that there will be binding of the active site head as well as the active site present on the actin filament. So these will form a cross bridge. Then after this ADP, finally another ATP molecule will come. So this will slide away. Again ATP will bind, hydrolysis will happen, and ADP energy is released, and then again this process starts. So there is a regular contraction and relaxation of the muscles. So let us see how this is happening. Basically, when sliding is happening, these thick filaments are as it is, but the thin filaments are sliding over it. So you can say that the I band is shortening or the sarcomere are shortening. You can see over here between these two Z lines, sarcomere is there. While contraction, they are shortening, but the thick filament remains as it is. Then afterwards, the calcium ions are pumped back to the sarcoplasmic reticulum, and again that relaxation happens. Now there can be two types of muscles. One is oxygen storing pigment in which it is present, that is myoglobin. Those are red muscle fibers, in which, or they can be called as aerobic muscles. But in other case where the quantity of myoglobin is less, they will appear whitish, called as white fibers or anaerobic muscles. So because this is activated in anaerobic process, and these help in during the aerobic respiration.